Hey, Mitchell Harmetz here. So, what are you? Well, you're you, obviously. Whatever your name is, your experiences, your DNA, you are you. But how do we define you? What makes you up? Is it your body? Is it your mind? Is it what you've been through? We need some type of way to represent this, what you are. And that's exactly what Sebastian Seung is working on. He has his own lab at MIT, and his project is called a connectome. Now what a connectome is a map of the brain. Think of it like a roadmap showing every single connection in the brain. You see, the brain is made up of neurons, and each of these neurons is like a tree, branching out at the end into things called dendrites. And each of these dendrites are connected to other dendrites in something called a synapse. Now this is what's really important. These connections are what the connectome wants to map out, because they're the sum of your experiences and your DNA. It's both nature and nurture. Your DNA provides the riverbed, and your experiences are like the water in the river. The riverbed provides the base, but the water changes it, shapes the river itself, just like your experiences and your DNA. Now, why is this important? Why should we know this besides the fact that it's pretty darn cool? If we could map out a connectome, a base human connectome, like we did with the Human Genome Project, a base human genome, then we could look for abnormalities, so to speak, in people suffering from neurological disorders, things like seizures, autism, ADHD, and see which neurons were firing too much specifically, which were not firing, where there might be damage, things like that, and really pinpoint these spots for treatment to help these people with these disorders. See, oh, during a seizure, these neurons are firing way too much, they won't stop firing, maybe with an electrical implant, which hundreds of thousands of people do have. We could target this specific area right here and fix this problem in a much better way. Stop this seizure. Alright, this is great. How are we doing this? How far along are we? Well, I'm happy to tell you we have the connectome of C. elegans. Now this worm here is C. elegans. It has around 300 neurons with 7,000 connections. These sound like pretty big numbers. We did a good job. Well, the human brain is suspected to have over 100 billion neurons, and who knows how many connections with that. So it's a little more difficult than this worm. But we're working on it. So you take a cube showing the neurons in that specific area. You color each neuron in a different color. And then you take another cube and another cube and another cube and keep coloring it in until you trace out a complete neuron in all of its connections. This takes a lot of cubes, and it's a lot of work, but it's being crowdsourced essentially. There are different programs online where you can look at cubes and color them in yourself, and that way millions of people can help out at once. I personally think it's going to turn into something like the Human Genome Project, where someone somewhere will have an amazing idea for how to do this in a much faster way. Now the next thing I want to talk to you about is a certain field of neuroscience called optogenetics. This was co-founded by Ed Boyden, who is doing some really amazing work. What it essentially is controlling the brain with light, causing synapses to fire or not fire using different frequencies of light, different colors. There exists a certain protein called chenorodopsin, contained by certain algae. When a certain frequency of light hits it, it opens a sodium ion channel. What this causes is a small electrical impulse to be sent in a cell. Well, the theory goes that Ed Boyden would locate the DNA that codes for this protein in the genome of the algae, extract it, replicate it, put it into a genetically modified virus. This virus would be modified to target specific cells in the brain of a mouse, mice before humans. And then this virus would then be injected into the brain of said mouse. These viruses would fly off, land on the cells they're coded for, inject the DNA, which would then be incorporated into these cells' genomes, and lo and behold, these cells would grow these proteins. This sounds really complicated. Well, because it is. So how close are we to it? How many steps have we gotten through? Well, we've done it. And it worked perfectly. These cells have grown those proteins. These cells can now be activated using light. That's absolutely incredible. One cell specifically that Dr. Boyden has targeted are basket cells. Basket cells are sort of like an off switch for a neuron. When they're activated, they shut down the neurons they're attached to. They basically cause them to stop firing. A practical application of this is during seizures. When a person is seizing, too many cells are firing at once and they're constantly firing. And these basket cells, which would normally be an off switch for them, are atrophied. They're damaged. They can't fire to shut down the overactive neurons. Now, if we could activate these basket cells using light during a seizure would stop. Basket cells would cause those overactive neurons to stop firing. The person would be fine. Another practical application of this technology is with PTSD. One thing he did was he exposed mice to a small shock, nothing really painful, just sort of annoying, when they heard a certain tone. So they began to associate this tone with the shock. Thus, when they heard the tone, even when there was no shock, they would sort of freeze up, stop moving, act sort of scared. This stimuli caused a scared reaction even though nothing bad was happening. What he did was he caused these proteins to be grown on the dopamine receptors, the cells which are responsible for creating dopamine. Dopamine is sort of like the feel-good chemical in the brain. You get it as a reward when you do something good. When he played the tone, he would then cause the dopamine receptors to fire. Thus, the mouse would get an influx of good feeling whenever it heard the tone. And sure enough, pretty darn soon, these mice stopped having that scared reaction when they heard the tone. Just continued on as normal. The mice which didn't have this, the control, they kept on acting scared. So this practical application really does work. Obviously, it's going to have to be changed some from use on humans and PTSD, but how it can be applied can really be expanded upon. So there's a form of blindness where the photoreceptors on the eyes are damaged. A very simplified manner of describing how vision works. These photoreceptors on the eyes pick up the stimuli, the light coming in. They pass it back, keeps on getting passed back all the way to the visual cortex, where it's then read and deciphered. Now, sometimes the photoreceptors are damaged. What he did was he caused these proteins to grow in place of the photoreceptors on mice. And these mice could detect light now, could essentially see. He cured blindness in these mice. The way he did this test was he had a 6-armed chamber. And there was a little water in the chamber so that the mouse couldn't really sit still. Only one of these arms was dry. And this arm that was dry had a light shining in it. These blind mice with damaged photoreceptors would just pick and choose randomly until they eventually got to the dry one. But the blind mice, which then had the photoreceptors grow, would immediately go to the lit up arm. These mice could see. It worked. It's mind-blowing. Now let's get into a little theory of my own. What if Sebastian Seung is successful? What if he makes a perfect connectome, a map of every single synapse in the human brain? We could then model this using a computer, create a 3D model of a person's brain, And then when exposing them to stimuli, we could see which neurons were firing at which moment in the exact order, and record this as a series of ones and zeros, 1 for firing, 0 for not firing. Now how an experience works is our neurons fire, basically telling us what's happening. Now if we turn these neurons firing into a bunch of ones and zeros into binary, have we not just turned an experience into binary? Because that's all that experience is those neurons firing. That's what we register. That's sort of scary. Now to take it even further, what if we combine this with Ed Boyden's research? What if we could then put these proteins on the neurons in the brain? And then could we not shine light of different frequencies in different areas and groups of neurons in the brain in the exact same order that was fired in the previous experience? And have we not just caused someone to have an experience they didn't actually have, because their neurons would fire in the exact same way as if they were having that experience? This sounds like mind control, and this sounds like something out of a sci-fi film, because it is. But this is research which could really lead to something like that, which is incredible, but it's also scary. It can be used in so many good ways, but it can be used in so many bad ways too. For example, say Alzheimer's, say someone is losing their memory, say they have amnesia. You could have recorded these experiences and just play it again for them, cause their neurons to fire in that specific way, thus they have their memory again. That's a great application. But what if you want someone to experience something they didn't actually experience? I don't want that. That's scary. No, my memories are my memories. I want to think I experienced what I actually did experience. Now this is unbelievable because it's taking something that sounds like some kind of conspiracy theory or something from a B science film, and it's making it in the real world. It's giving it away. This could actually happen. Now, is it actually going to happen? Probably not, because a big thing in science is ethics. Is something ethical? Should we be doing this? In order to get the money for funding, it has to be approved, and that's where ethics come in. Additionally, we need things like supercomputers to perfectly process the firing in the exact order, and a perfect light source to fire in the exact area. But these are things which could come in the future, these technologies. Another possible flaw in this is that each human brain is different, each connectome is different with different neurons, though there is probably a base connectome. It's just a theory, just like anything else is. Now the real question is, how do we know this hasn't already happened? Is there a way to know? Am I just hooked up to some amazing light source to a supercomputer? I don't know. This research is saying it may be possible. Who knows? And as always, thanks for watching.